Okay, so for the interest of time, we'll get started. So welcome again, everyone. My name is Andrew. I'm a scientific consultant for NIRx, and it really is my pleasure to be joined today with Julie, and who'll be talking to you about the LIONirs Toolbox. So the title for today's webinar is Introduction to the LIONirs Toolbox, a freely accessible NIRx data analysis tool. So if this is your first time joining one of our webinars, of course, welcome. Just to tell you a little bit about us. So NIRx has been developing cutting-edge NIRS equipment for the last 20 years or so. We've been focusing on researchers with training and outstanding support and providing a lot of collaborative environment. And this webinar is just one of our efforts towards doing that. Our solutions are developed in Berlin. However, they're distributed worldwide by our team of distributors. And we also have scientists to support all of our users. So if you'd like to connect with us, you can reach us on social media. We're available on various platforms. You can also subscribe to our newsletter, where you hear a lot about our featured products and, of course, some of our exciting news from our users. We promise not to bombard you with lots of emails, so you only get 3 or 4 emails from us a year. So joining me today from our wonderful admin team, Alina and Cami, and they're really helping make sure everything runs very smoothly behind the scenes. And a couple of housekeeping points before we get started. So everyone on this call is muted. Questions are welcome at any time. You can use the Zoom chat function. We will hold all the questions until the end of the webinar, and that's when we'll ask Julie. Of course, if you prefer asking the question directly, you can always use the raise hand function in Zoom, and then you can unmute yourself and ask your question again at the end of the webinar. The content, so this webinar is being recorded and will be available online in the upcoming days. And if we don't get to all the questions today, please feel free to email us at consulting@nirx.net. And of course, if you're an existing user and you have any questions, you can email us at support@nirx.net. So without further ado, it's my pleasure today to introduce Julie Tremblay. So Julie is working as a biomedical engineer at the St. Justine Pediatric Hospital in Montreal. And in 2007, or since 2007, she's providing a lot of technical assistance in programming and data analysis with lots of projects focusing on multiple neuroimaging data analysis, so including NIRs, fMRI, DTI, and EEG, to name but a few. She also had the opportunity to develop several tools over the years to facilitate and automate analysis for projects at the Lion Lab directed by Dr. Anne Gallagher, and recently was able to integrate all these wonderful tools in the LIONirs Toolbox. So without further ado, I would like to welcome Julie, and please, everybody, enjoy the webinar.
Thank you very much for the introduction. So I share my screen to present you the LIONirs Toolbox today. So it's a thank you for inviting me at this webinar. It's a pleasure to present you my work in the LIONirs Toolbox today. I'm working in St. Justine Pediatric Hospital, as Andrew told you. We are using the infrared spectroscopy on many research projects, mostly related to children development. And some with various neurodevelopmental conditions. This population is challenging to test. So the aim of this toolbox was to give us flexibility in our data analysis, and we are happy to share it with the community. It is now freely available on GitHub. Today, I'd like to give you a quick introduction about the toolbox we published last year. At the beginning, we designed it for only the goal of the toolbox was to give us the ability to follow the data during the analysis and to explore several methods in parallel and verify the localization of the result using a 3D visualization. We test many children in our lab, and this type of data could be highly artifactive. It is therefore worth it to try to perform artifact data correction, such as target PCA or Parafact, to keep statistical power, but also to reject data that you don't trust and can't be corrected. We definitely want to avoid a black box phenomenon with this type of data. The quality check is performed at the channel level and could be specified for a short interval of time. The toolbox allows you to perform standard analysis on the hemodynamic measurements, such as averaging time course or using a general linear model approach. Few connectivity measures are implemented, such as coherence in the frequency domain or person correlation in the time domain. It is also possible to export your data and to continue the statistical analysis in a specialized software of your choice if needed. So the first part to define when you design an experiment is the way you will install the source and detector over the cap. So we like to register the object over the head to ease the interpretation. We perform the tissue segmentation in an image processor software to edit the MRI tissue and create a surface representation of the skin and the cortex. So this procedure could be laborious and individual MRI are not always available. So pre-segmented templates from children to adults are available if you need them. So when it is done, the spatial coordinates of the cap are registered with the reconstruction. Small adjustment for the coordinate registration with the MRI anatomy could be done manually, such as rotation or translation. If you need, as example, a small rotation of the MRI could be necessary if the cap were placed in the bottom or the top of the front end. So we measure the cap position during the NIRx recording to ensure good consistency among our subject and take picture of the installation to be sure that the registration represents the reality of the recording. Once this reconstruction is performed, you can read the data from the NIRx recording file. Analyzed data file will be saved in a folder organized by subject. Other popular format data such as SNIF or .NIRx already integrate the spatial coordinates. So the data could be read directly using the embedded coordinate, but you will need to add the MRI surface if you wish to use it. So people familiar with MATLAB and SPM Toolbox will find the LIONirs easy to install. The Toolbox is developed using the SPM batch editor, mainly used for fMRI analysis. Once the Toolbox is installed, you need to insert the operation to build your pipeline and customize the option according to your experiment. The Toolbox will apply the operation sequentially. So to know what an operation do and how to use it, you could refer to the user manual or the short description in the bottom of the batch editor. Once you are satisfied with your pipeline, it is a good idea to save it, to apply it as a template for other data sets. It is recommended to add the display GUI at the end of your pipeline to visualize the data after the operation are performed and make manual adjustment on the current step if needed. So it's a good idea to keep an eye on the command window to keep track of the operation performed. It will write you which new analysis file are created or if potential error occur. So the display GUI, as its name mentions it's a visual interface used to display the data. It opens the data structure and allows you to visualize any step of your data, but only to modify the current step as the operation are performed sequentially. The artifact could be reject or decompose for a specific time and channel by using the right click on the data window. You can select interval and channel where you will wish to apply your modification. You select the decomposition method to apply, such as PCA, Parafac, or GLM, to extract it for selected time and channel. Once you get the component related to an artifact, you may subtract it. If you get a component related to a task using GLM or average, you may export it for further statistical operation. If you have the opportunity to record data such as video, EEG, or external physiological measurement, you may integrate those in your data. However, to integrate multimodal data, you must ensure to synchronize them with NIRx by using common trigger at the acquisition and segment this marker in your pipeline. It is also a good practice to test the synchronicity. If you use them over a long period of time, a time lag could appear, so it could be a good idea to use several markers to detect time lag and do your validation. It will be possible to open the EEG for a specific time window if you integrate it in your data. And you can get the video corresponding to this event if you enter it, or if you enter auxiliary data, they will be available for visualization in the upper window. It will allow you to project the data. Here you can see it's the respiration bell that we measure. And if you press on the 3D montage, it will allow you to visualize the data over the topographic map. You can project over the anatomy using a simple projection or using an inverse weighted average projection. It also helps you to localize your channel selection and to create region of interest. So here is an example of data recorded with a 7-year-old boy who is performing a verbal fluency task aloud. So I take this example of data to try the semi-automatic artifact correction because this data set are full of muscular and movement artifacts as the young participants do not stay immobile for a long period of time. The hemodynamic signal is a slow process, and we need to record a stable signal during enough time to measure the slow variation. On that recording, only 2 segments over 11 show a signal without perturbation. So we perform the automatic artifact detection, which is based on a roll average to detect abrupt variation in the data. All events where a perturbation is detected are marked in yellow. Subsequently, on this yellow interval, the parafact decomposition was applied automatically to isolate the influence of strongest component, generally caused by movement, and subtract this component from the data to minimize their impact. So this is what the correction looked like after the automatic correction. Finally, we decide that we were happy with this correction and we keep all the events for our analysis. It's also possible to keep them marked in yellow if you wish to reject it from your data. How an artifact segment could look like on the hemodynamic time course. So in red here, you can visualize the artifact in the raw data and how it looks like after the transformation in hemodynamic concentration. We do observe a strong variation where the HBO and HBr concentration increase strangely together. We like to avoid this on her final data because it was caused by a 2-second length artifact clearly visible in the raw data and not relate to our task. So we observe that time course vary together. So we really don't want to integrate this in our data. After the correction applied, it greatly improved this segment. The raw intensity looked almost unperturbed by the artifact after the correction. In the hemodynamic corrected in black, we observed that the slow increase well synchronized with the 15 seconds of the task performed by the subject, just here. So it's more meaningful for us that looking at the data corrected. It is important to keep in mind that data decomposition are helpful, but need to be kept in the context of the data. If they are available, it's always good to use the video and EEG to understand what's going on and keep note of your recording. As we just observed, the uncorrected data could greatly affect the outcome of the analysis. In the case that the contact with the skin or the data are of poor quality, it's better to consider to reject this part of data. But in the case of small and discrete artifactual event occur, they have a good outcome with data decomposition method to be able to correct it. So be careful to overcorrection. It could also subtract the activation itself. And remember that a good acquisition is always better than a correction. So I like to give you a short mathematical review of the data decomposition technique. PCA techniques aim to decompose bidimensional data to explain the data in few components. The first component are selected to explain the maximum amount of variance in the data, and subsequently for the other component. Each of them will be defined by orthogonal vector. In our case, the data will be organized in 2 dimensions by time and channel dimension. Usually, the artifact we aim to decompose will be orthogonal, as artifacts are often marked by a strong variation in amplitude, discrete in time, and visible in surrounding channels. So PCA perform well, and it's easy to apply automatically by taking the first component and subtract it. But in some cases, the orthogonality assumption is not respected. As example, when multiple artifact sources occur during the same period of time. In that specific case, parafact could present an advantage. The parafact decomposition use multiple dimensions and do not constrain orthogonality as PCA. So the method was initially developed for chemical application to distinguish the fluorescence spectrum of different materials. So to decompose parafact, we organize the data in 3 dimensions using the temporal, the spatial, and the wavelength information related together. As shown in our previous example, we observed that both wavelengths are generally affected together by the artifact. So it's an advantage to decompose the wavelength conjointly. If we compare the correction between parafact and PCA for the same artifact, so we select the interval and the channel to perform the decomposition. As both methods are sensible to outlier, it could be good to remove channels that need to be rejected in reason of the acquisition. The way you select the time and interval will influence the decomposition. So it's usually helpful to target the decomposition for distinct events. On the left side, you could see the example where parafact was subtracted. On the right side, the first PCA were subtracted from the data. Both methods greatly reduce the artifact, but parafact could present a small advantage in specific case. If you want to know more about comparison between artifact correction method, I invite you to read our paper published in Neurophotonic last year. So here is a specific example where the subject scratches his head on the video. So we like to show how the decomposition could capture the complex time course of the muscular and cap movement occurring during that interval in few components. So the slow time course with the cardiac artifacts seem to be well isolated too. So this part is a bit technical, but important to understand if you wish to use the toolbox for your analysis. As I mentioned previously, the SPM batch processing applies the operation in a sequential workflow. So at any step, you can stop this workflow and visualize your data in the display GUI to review and eventually modify the artifact detection. For some analysis design, it could be advantageous to create parallel pipeline from a specific step to create a nested structure with several branches of analysis. It will allow you to see the impact of several pipelines, to compare them, or simply to process different conditions. So the tool new branch will be used to create subfolder in your analysis tree and allow you to start several pipelines from the same node. So here we could follow our example from the young boy data we correct previously. We try both pipeline, the average of the hemodynamic time course for the uncorrected and corrected data. On the uncorrected signal in black, in red, we can see that the standard error is large and the activation very difficult to detect. So in contrast, the corrected data in black, the average activation is clear and the error bar are smaller. So reducing the variability caused by artifact greatly improves the chance to get meaningful statistical measure. So a common way to analyze NIRs data is to estimate the hemodynamic response using multiple linear regression. Here is an example of how the GLM could be applied on NIRs data, where the hemodynamic time course will represent the variable measure in Y. We estimate the coefficient for the regressor in the equation. So we define as regressor the HRF response convolved during the period of time of our paradigm. And as a second regressor, we use our short distance channel to model the physiology. In fact, the rule to define the GLM in the toolbox are really flexible. You could use as regressor any auxiliary data previously defined in the upper window and select the short distance channel as you wish. So it may also be a global regression using out channel if short distance channel are not available. It will give you the flexibility to explore the GLM model in some meaningful way for your data. So when you perform the GLM analysis, an estimation of the coefficient will be saved for each channel. You can do statistic on this value at the subject or the group level and open your result on the topography. Basic statistics are available in the toolbox, but to perform more complex analysis, it is better to export and perform them in specialized statistical software such as R or SPSS. When you perform and save the value by channel, you could reopen it in the 3D montage to visualize the statistic in a topography. So the toolbox can also calculate some connectivity measures such as person correlation. The correlation measure the relation between the time course of two spatially distinct channels. So each seed in the connectivity organization could be defined at the level, channel, or a region of interest. So in the option, we implement person correlation to be applied as a circular bootstrap in the data and get an average measure of the connectivity. So the connectivity measure come from many random samples and exclude the part where artifacts have been marked. So it allows to get a connectivity matrix that is gets stable when they have enough segments. So we inspire this approach from a method described by Belek and al. And apply in fMRI connectivity. So to organize this connectivity data in some meaningful information, we define a region of interest in the 3D montage by selecting the channel closest to each area of the 1020 system. So each color will correspond to a region defined for each hemisphere. You could customize your region as you wish. It could be based on an atlas. It's just to select the channel that and organize it in a meaningful way. Once your region are defined, you need to select the order in which you wish to display the region. As example, where we organize left and right hemispheric information in the connectogram, and the matrix start from frontal to parietal. It is possible to view the connectivity data as a matrix. So you can observe on the matrix a strong connection in the frontal area as well as a cluster in temporal area. Each correspond to functional region organized together. So some inter-hemispheric connection are also present. We like to display the connection in a connectogram shape to get a quick overview of the region connected. So once again, you can organize the region as you wish and adjust the threshold for visualization of the link, and some statistical functions are also available. So we recently published our paper in the Journal of Neuroscience Method. So if you're interested to use the toolbox, it's a good way to get introduced. You can download the code on GitHub, or you can also find a link on the NIRx website. All the code was developed in MATLAB, so you will need to install MATLAB and the Statistical and Machine Learning Toolbox. So you also need to install SPM and to copy the LIONirs in the SPM Toolbox section to add the LIONirs in the menu of the batch editor. So you just have to specify the path in MATLAB, and it should work in all the recent versions of MATLAB. In the wiki page, we provide few data examples of analysis, segmented MRI, to help you to get started with the toolbox. It's compatible with NIRx data format. We also do some import function for .NIRs, the one compatible with Homer and the SNF format. It's also possible to export in those formats if you wish to continue your analysis in other toolbox. I like to thank the Lion Lab teams and collaborator for all the fruitful discussion and problem we try to solve together, and also for the financial support we receive for our research. So I hope it could help you in your data analysis and to have the opportunity to improve it according to further needs and research questions. So thank you for your attention. I will be happy to answer to your question.
Thank you very much, Julie. That was wonderful. Thanks so much for the overview. So the floor is now open for questions. If anybody has any questions, you can type it in the chat. You can also use the raise hand function, and we can call on to you to unmute yourself and ask your question. So I'll get us started, Julie, with a few questions that came in. So a couple of questions that came were asking about how to install the required software and if there's any training videos. Now, you did hint on it, you know, what you need SPM, you need MATLAB, but are there any other resources? Do you have any training videos up that people can refer to? Of course, this webinar, they can refer to it, but are there any other resources out there?
I think the good place to check it's in the wiki of the GitHub. So I started slowly, but I include several data examples that you can run with some batch inside, and they show you where to click and where to how to use it. So I think that's a good way to start with some working data and after to try with your own data.
That's perfect. And sorry if I missed this, was there a specific version of MATLAB that you needed, or?
Our version seemed to work well. So I tried with the last one, I tried with the 2018, I test. So basically, I'm developing on Windows. So I test more for Windows, but as it's portable, it's possible they have some small problem to adjust in other environment, but mostly it should work on all of them.
Awesome, that's great.
It's to test it.
Yes, absolutely. I see a couple of questions in the chat. So the first question is, what is the recommended data length for resting state functional connectivity using NIRs with a sampling rate of 20.3 Hertz?
Sorry, could you repeat the question?
Yeah, so it's one of the questions in the chat. So the question is, what is the recommended data length for resting state functional connectivity using NIRs with a sampling rate of 20.3 Hertz?
Yeah, us, we try to have as much as possible, but at a point, as we capture the slow signal, we try to have at least 1 minute stable. And as I told you, we do like a bootstrap, so it's helped us to get like the best part of our data. So we use random sample during the 12-minute recording. And at a point, we really see that it gets stabilized and the matrix doesn't change much.
Perfect. And another question that came in is, does the toolbox help users to decide opto placement? I understand it will help with virtual opto placement on the brain or head model specified.
No, we don't have that. I think that NIRx are some package for doing it, but us, we're more placed in logical organization, like we're looking where it's go over the skin or cortical representation and ensure that we have good coverage over that area, but it not help you to place the optod. One of the hard thing is to keep consistent in your recording, like having always the same optod placement will help you. So from recently, we try to keep standardized montage to have the much coverage as possible.
Perfect. And a question about the features. So I see there are two questions. We'll kind of combine them. One is, does it support, is there any functionality for hyperscanning? And the other question was about real-time signal pre-processing.
Not yet. It's really post-processing. It's not as in the recording software. So once you record your data and you want to have a closest look on that to deal with it, is just post-processing software.
Perfect. So the users are able to essentially extract the data and analyze it themselves in MATLAB, kind of even so pre-process it and then maybe do some extra analysis.
In the NIRx acquisition, you have some averaging that are online. It's good to have a look at it when you record, just to ensure you have enough data, but you cannot do the artifact correction or deal point by point. So
perfect. And I see there was another question. Can the toolbox be used to deal with artifacts induced by current TMS? So maybe you can expand a little bit on the sort of procedure that you use, right, for artifact removal, and does that apply to artifacts induced by TMS?
I don't know how, maybe this question have to be precise, because you talk, I don't know what looked like the TMS artifact on NIRx. It's probably, if it's a distinct artifact, it probably could be removed using data decomposition method. It's to try it.
Okay, perfect. And yeah, sorry, go ahead.
I do some tries. So we have a data set with EEG that have a cardiac artifact, and with the target PreCA, it was able to remove it very well. So we can save this kind of data, but it's to try. Actually, it's I don't know if it will work perfectly for that kind, but it's to try it.
Awesome. And along the same lines, while we talk about PCA, is there sort of a recommended minimum number of channels that you need for the PCA to work well, or can it work with even a simple, sort of 8-channel, or do you need a large number of channels? It's
work with a large number of channels, but as I told previously, as it's orthogonal, sometimes if you have very two distinct patterns, it could be useful to distinguish. Like if you have few artifacts that are very flat and do not correspond to the rest of your data, sometimes you will get better decomposition by rejecting those from your decomposition.
Awesome. I see another question that came in. Is it possible to implement additional motion artifact correction methods such as wavelength or spline interpolation?
Yeah, probably it's just the batch editor. It's easy to add new function. It's just to add a model with what you want to add. So probably in the future that can be a good idea. But for yet it's not available.
Perfect. And about features as well, you did mention that you can integrate other, physiological measures or other sort of modalities as well in there, as long as everything is triggered properly. So a question that came in about that as well is, does it support regressing short distance channels at the moment?
Actually, as I include in the GLM, you can use auxiliary or channel in your data. So if you have your short distance channel, you treat them in your recording as other channels. So you will perform the pre-processing and transfer in concentration, and you can add it in the GLM if you wish to subtract it.
Perfect. I see a couple of other questions. So one is about how can you compare it to other toolboxes that are available, Homer, NIRx Toolbox, and perhaps even comment about can you integrate some of the stuff? So you pre-process in one, can you incorporate, the next result into the other toolbox? Does LIONirs allow that sort of integration at the moment?
Actually, what we really focus, I think the advantage is the online visualization of the artifact and the flexibility to interact with it. So, and to select the channel and interval for your decomposition. You can do part of the pre-processing using the LIONirs and export them to continue in other software if you're used to. It's just, I think that's the advantage. And people can use it as they wish to use it. It's
Perfect. Another question is it's more about FNIRs in general, and it's about how fast is the detection of oxyhemoglobin and deoxyhemoglobin variation during a test or an experiment with FNIRs. So I'm not sure if this question is tailored more towards real time, but it's just a general FNIRs question that we
Actually, it's an interesting question, because we know that the hemodynamic in theory, like peak after 5 seconds and have his plateau for the time of the experiment. But when we look at the data time course for a project in epilepsy, we really see that sometimes it starts from a place and it's spread to the surrounding area. So it could be interesting from 2 or 3 seconds, we really see the start of the hemodynamic signal starting from a place and slightly affecting the surrounding area. So it could be interesting to check. As in this, we have a high time resolution. It's a paper we published in epilepsy with Dr. Nguyen, and it was about frontal lobe epilepsy. So we really have a look at the time course and see that in the first second, it's interesting the effect you can observe in the hemodynamic dynamic.
Perfect. Yeah, we'd love to also get this paper from you and include the citation as well, so we can share with everyone that's attending. So 2 other questions. Is it possible to add an alternative for short channels such as spatial domain filtering? And can you elaborate a little bit more about hyperscanning as well? So elaborate a little bit more as an option. So you mentioned it's not supported right now, but you can do it externally. Is there any sort of tips that you have? So I guess that's one question. The other one, can you add an alternative for short channels such as spatial domain filtering?
I do not work for hyperscanning now, so I don't have magical answer for this. I don't know yet for this question.
Okay. And in terms of adding something other than short channels as a regressor.
Yeah, I like to use the global of all channels. When it's a short distance channel or not average, like taking all the channel, I think that Zhang published a paper using the old channel instead of short distance to regress them. And it's we compare result with short distance and global, and usually if you do the correction, both work and give similar results.
We have a question here about, does the toolbox support MVPA? So I'm guessing that's multivariate parametric analysis at the moment.
No, we mostly do using channel. That could be do maybe for topography or stuff like that, but yet it's not supported. The best I do for that is trying to do cluster as looking for surrounding channel and try to create region of interest for surrounding class channel that are significant too, such as in the field trip. They have nice method to do it, but it's not support the MPVA.
Great. And there was another sort of general question I had about resting state and connectograms. I really like the figures you showed. Those are a lot easier to look at, right, compared to looking at like a matrix in general. Do you have any recommendation for thresholds to use when you get the resting state? The connections looked very clean and very nice. Is there a threshold, or is it participant dependent?
I think more participants you have, the clearer it gets. As when you look subject by subject, it's not always that clear. But basically the threshold are often like by using statistics. You look at the significant link and
Perfect. So some sort of like permutation analysis in a sense to find.
Exactly.
Okay, wonderful. I'm just keeping an eye on the chat to see if there are any other questions. Okay, I don't see any other questions. Of course, if anybody has any other questions, you're welcome to email us at consulting@nirx.net. We will pass the questions over to Julie, and we will make sure to get back to you with answers. So if there are no further questions, thank you everybody for joining us. We are actually right on time as well. So Julie, this was wonderful. It was great having you. Again, the recording will be made available in the upcoming days on our website. And with that, thanks everyone, and have a wonderful rest of your day.
Thank you.